Okay, hello everybody. So this video is going to be all about microscopes. And so that is going to be the title of our notes today. Okay, so microscopes are a tool that biologists or scientists in general use to look at very little tiny things. Okay, so we see things, and part of science is making observations, and there are some things that you actually cannot see with the naked eye, but the microscope opens this whole world of little tiny organisms that we can see. And so I first want to start off by telling you that the first microscope was designed or invented by a scientist named Hooke in 1665. So in 1665, so a long time ago, Hooke made the first microscope. And so what happened was he was just a regular guy. He wasn't a scientist. But his hobby was making lenses. Okay, that was his hobby. Is he would make glass and he would make lenses, and it was just his hobby. And so he would make better and better lenses to the point where he could see these little tiny things. And Hooke was actually the very first person to see microorganisms. So first to see microorganisms. And when I say microorganisms, what I'm talking about is bacteria. And so what he did was he started looking at everything, and he would see these little tiny things moving around. And he even named them animalcules because they look like teeny tiny animals. But what he didn't realize what it was actually bacteria, and that bacteria is everywhere in the world. We just can't see them. And so he even scraped off some of his tooth and he put it in a slide and he looked at it under the microscope, and he was the first one to see, like, oh my goodness, there was bacteria on my teeth. So he was the one who is like the founding father of microbiology, but it was like many, many years later that we like figured out more about bacteria. But I want to point out that using a microscope allows us to see little small things. And the microscope that we use in our class is called a light microscope. And so I'm going to show you a picture of a light microscope. And so this is the microscopes that we will use in lab. And sometimes it's called a compound light microscope. Okay, so here's a picture. And it's called a compound light microscope because there are multiple lenses that reflect the light. Okay, so let me explain. So what I'm going to do is, here's a picture of one, but we are also going to draw one together. Okay, and then we are going to talk about the path of the light that goes through it. So let me scoot up a little bit. I'm going to change colors to blue. And so as I explain the path of light, I'm going to also explain all the different parts of the light microscope. Okay, so in light microscopes, you have to have a light. And so here is a light source. I'm just going to put it on the base of a microscope. And so we're just drawing one. This is not going to be like art. This is just going to be like a biology notebook drawing, okay, which is not art. And so what I want to put here is I'm going to label them in red. I'm going to label them each part. And so this is going to be a light source, okay, which is why it's called a light microscope. Okay, there are other microscopes, which I will talk about in a second, that don't use light. Sorry, that's kind of messy. And so there's got to be a light source. And so in this picture, here is the light right here. It's on the bottom. So you have to plug it into an electrical source, and there's usually like an on-off switch for the light. Sometimes it's a nozzle like this where you click it on and then you can adjust how much light is going in, like a lot of light or not very much light. You can adjust that to make a better view of what you're looking at. Okay, so I'm going to talk about the path of this light. So I'm going to draw the light as pink, just so we can like have different colors here. Okay, so the light goes up, and where it goes next is onto the stage. Okay, so we're doing a blue microscope. And so I'm going to draw this next thing is called a stage. And in our picture here, you can see the stages right here. Okay, so it's just like a platform. And what you put on the stage is you put a slide with a specimen on it. So I'm just going to draw it like this. So here is the specimen. Okay, normally you put it in a slide. The slide is made out of glass usually. So I'm going to put specimen. And what that means is it's what you're looking at. So it could be, a sample from your skin, or it could be a sample from a leaf, or just anything you want to look at. Okay, so normally what you do is you have a glass slide, you put the specimen on the glass slide, and then you put like a little cover slip over that. Okay, and maybe some water in between just to keep it all very flat. And so that's what your specimen is. It is what you're going to be looking at. The specimen has to be very thin. It has to be thin enough for the light to pass through it. If it is too thick and the light gets blocked, then you're not going to be able to see it. You're just going to see like this dark blob. So it has to be cut very thin so that light can pass through. Okay, so that's one thing. And so in the lab, we will make some of our own specimens. And sometimes you can buy them already made. Okay, so the path of light is going from the light source through the stage, and so the stage has a hole in it, and then there's a specimen on here. Okay, another thing that I want to point out is underneath the stage, there is what is called a condenser. Okay, so this is called a light condenser. And so what happens is you have all this light, and usually it's kind of scattered light. Okay, so it's going in all different directions. So I'm just going to put arrows here. And the condenser will narrow the light so that it's in one straight beam. Okay, and that's important so that you can see your specimen better. So there's a condenser on the bottom here that's going to narrow out the light, and then you can see what you're looking at. Okay, then here you can see on the picture of the microscope there are these lenses, and they're like these little tubes that come out of this area that's called the nose. Okay, so I'm going to draw a nose here. So let me label this nose. And then coming off of the nose, you have these different tubes which represent lenses. Okay, and these are called objective lenses. There's usually more than one. Sometimes there's just one. The ones we have more than one, and this one in the picture has 4. Okay, objective lenses. And so usually with the objective lenses, I'm going to like put an arrow over here just to talk about it a little bit more. There's always like a low, a medium, maybe another medium, and a high. Okay, so the low lens is usually called a scanning lens. And the low lens can be anywhere from like 4x or 5x. Let's just put 5x, okay? And then you have a low lens, and let's say that's 10x. And so every microscope is different, okay? So this is just like an example. So depending on what microscope you have determines what the objective lenses are, but this is like in general. Okay, then we have a medium lens, and sometimes that is 40x, and then you have a high lens. Okay, and that is 100x usually. And so depending on how small your specimen is determines which objective lens you're going to use. If it's something that's kind of big that you can kind of see with your naked eye already, you can just use the low lens. If it is something really, really small, like one of your cells, then you're going to have to use the high lens to see it. Usually the scanning lens is used just to locate your specimen. So it's just this kind of this like, let's just find where the specimen is, and then we're going to use the low, medium, or high to then narrow in to get a really good picture of it. Okay, so usually you don't look at things under the scanning lens. It's just to help you orient yourself so you can find your thing that you're looking at. Okay, so those are the different lenses. The nose rotates and it clicks into place when you get to the objective that you want. Okay, so if you're looking through the microscope and it looks black, it's because the objective lens isn't clicked into place. Once it clicks into place, then the light can pass through. So I'm going to draw the light here. So it gets condensed, it goes through the specimen, and it goes through the objective lens. And then inside the nose, there is a little mirror. Okay, so I'm going to draw that here. And the light is going to reflect off that mirror. And then we usually have what we call a second lens, which is called an ocular lens. Okay, so let me label this. This right here is called the ocular lens. And it's called that because this is where your eyeball is. This is where your eyes look in. So here we have two. And so some microscopes only have one. If there's two, usually you can adjust it because everybody's eyes are like spread apart or close together differently. And so usually what I do is I'll just like cover one eye, just use like my dominant eye to look in it. It's hard for me to look in using both eyes. I don't know why, but it's just what your preference is. So the light is going to go from the light source. Okay, actually, this is supposed to say light source. Sorry, I gotta erase this. Not light microscope. This is a light source. So it's gonna go from the light source, and then it's gonna go through the condenser, through the stage, through the specimen, into the objective lens, whichever one you choose. It's gonna bounce off the mirror that's in the nose, and it's gonna go through the ocular lens to your eyeball. Okay, the ocular lens is a second lens, which is why sometimes this is called a compound light microscope. Compound meaning there are two lenses. We have the objective lens and the ocular lens. The ocular lens is always 10x, okay, magnification. Okay, and then I'm just going to draw, this is where your eyeball is. So this is your eye, okay? Let me just make this like a nose and some lips, and here's the neck. So just to give you a reference, here, let's put some hair on this person. Okay. So that is a compound light microscope. For you to know what the magnification is of what you are looking at, what you do is you multiply the objective lens to the ocular lens. So if you are looking through the scanning lens, so let me just write this here. So I'm going to say total magnification. So this is to calculate your total magnification. Oh my goodness. Sorry, this is kind of messy today. What you do is you multiply the ocular lens to the objective lens. So I'm going to put ocular times objective. Okay, so for example, the ocular is always 10x. So I'm going to scoot up a little bit here. And the objective depends on what you chose. So let's say you're looking at the scanning lens. So let me just put scanning. And your scanning lens is 5x. So that's going to be a total of 50x magnification. If you're looking at the low lens, that's going to be multiplied by 10x. Sorry. And so that's going to give you 100x, which means the thing you're looking at is 100 times bigger than if you looked at it without the microscope. So that's what that means. Okay, so then if you looked at your medium objective lens, that is 40x. So you're going to multiply 10 times 40, and that's going to give you 400, which means you're looking at it 400 times bigger. And then the high is going to be 100. So that's going to be 1,000 times as big. Okay, so usually a compound light microscope allows you to see something up to 1,000 times what it normally looks like. And so the word that we like to use is resolution. So the better the resolution, the better the microscope. Okay. And so the more powerful a microscope is. So I just want to give you this vocabulary word. Resolution. So the higher the resolution, the better you can see the object you're looking at in your microscope. And what a resolution means is that you can see two things as, okay, let's say we're looking at two things and you're looking at it without the microscope, it would look like it was just one thing because they're too small for you to determine that they're actually two things. So resolution means that you can see objects that are close together as separate. That's what it actually means. So I'm going to say that to see objects that are close together as separate. Okay. And so the more magnification you have, the better the resolution is. Okay. There is another trick with microscopes that we use for better resolution, and that is we stain the specimen. Okay. So for better resolution, sometimes we stain the specimen. Okay. And if you think about it, let's say you're looking at a cheek cell. So in the lab, sometimes one of the labs is we'll take a Q-tip and you'll scrape the inside of your cheek, and then you'll put it on a slide and you'll look at that in the microscope. But first we're going to add like a blue stain to it because your cheek cells are usually clear. And so if you're looking at a clear cell on a clear piece of glass, it's going to be very difficult to see it. But if you stain it blue, then now the blue is a contrast to the clear, and now you can see it. Okay, so that's like one of the tricks that we use when you do microscopy, is what it's called. So what I'm going to do is I'm going to show you a picture of a cheek cell in a light microscope. And so this is what it looks like. So these are stained cells, and so they were scraped off a person on the inside of their cheek. And each of these cells, they're all squished up on top of each other. And cells are like that sometimes, like some cells are like on top of each other like this. And so you can see that there's like this kind of blob shape. So this is one cell here. And this little dark spot in the middle is what is called the nucleus of the cell, which we'll talk about in a later video. But here's a second cell here. It's like a blob with a nucleus. Here's another cell here with a nucleus in there. And so you can see 1, 2, 3, 4, 5, 6 cells in this specimen. And you can see the 6 nuclei. Okay, so that is what a compound light microscope is. And okay, and so I just want to talk about a different kind of microscope. And this is a more powerful one. Okay, and this is called an electron microscope. These were invented in the 1950s, so not that long ago. So I'm going to put a title, electron microscope. And so these were invented in the 1950s, like when technology had advanced a lot. 50s, invented. And electron microscopes don't use light, but they use electrons. And we've talked about electrons in a past video when we were talking about chemistry. And what happens is electrons are shot into a tube, and electrons scatter and they create an image of what they're being shot at. Okay. And so they're very, very expensive. Very rarely does a college have one of these, but sometimes they do. And they're very difficult to prepare. Like the samples are difficult to prepare. You have to slice them very, very thin, and you have to do all these things to get them ready. But once you do it, you get these really amazing pictures. And so I just want to share some pictures with you of what electron microscopes can give you. So here is a picture of cheek cells in an electron microscope. So we saw them in the light microscope, and this is what they look like in an electron microscope, which is way more detailed. So here is what your cheek cells actually look like. So I'm going to write a label here. Oh, okay, so these are cells, human cheek cells. Whoops. Okay. And so let me show you some more pictures. Here is a picture of the head of a fly. So that's very large. And so this is what a fly head looks like. And so, like I said, you can get a lot of incredible detail using these kind of tools. And you could see a fly head in the microscope, but you wouldn't get this amount of detail. So you could see that their eyes are made of all these little tiny cells. It's really cool. Let me show you some more pictures. Here is a picture of a bed bug. Yuck. Look at that. That's just crazy and gross. And then I want to show you one more picture of a cell undergoing a process called apoptosis. So this is a cell in the lab. And so it's actually a cancer cell and it's undergoing apoptosis. So I'm going to write this down. Just put a label on here. Cell undergoing apoptosis. And what apoptosis is actually like a cell committing suicide. So sometimes your cells get, in trouble, and so they'll just commit suicide to protect like the rest of the body. And so this process called apoptosis, we will talk about this in the future, but I just want to show you a picture. So usually cells are very round, but this cell's like under destruction right now. So it's pretty crazy. Okay, so the last thing that I want to talk about is that because of the microscope, we can see cells. And so cells are the basic fundamental unit for life. Okay, so this class is all about biology, which is the study of life. So I'm going to write that down. So biology, the study of life. And the fundamental unit of life is a cell. So let's write that down. Is called a cell. Okay. And so if you are looking at a living organism like a human, like me, the basic part of me are cells. And that's what we're going to be talking about this week. And so there is this theory called cell theory that has some rules. Okay. And so we're gonna write down those rules. So cell theory was able to be developed because we had microscopes and we could see cells. Okay, so let's see. Cell theory states the following three rules. Okay, rule number one is that all living organisms are composed of one or more cells. So all living things, all living organisms, if something is alive, it is called an organism. All living organisms are made of one or more cells. Hang on, let me scoot over just a little bit. And I want to point out that the bacteria that Hooke saw under the microscope are organisms that are made of just one cell. So there are unicellular organisms that are just made of one cell, and an example of that is bacteria. We are made of trillions of cells. Okay, so a lot of like the animals that we think about are multicellular, made of many, many cells. But all living things have to be made of cells. If they are not, then they are not alive. So, for example, a rock is not made of cells, it is not alive. The sun is not made of cells, it is not alive. Water is not made of cells, it is not alive. But plants are made of cells. Bacteria are made of cells. Mushrooms are made of cells. We are made of cells. And so cells are what make up all living organisms. Okay, so number two is that cells are the basic unit of structure and function. So let's write this down. Cells are the basic units of structure and function of living organisms. Structure and function. So that means the shape and the physiology, like what makes you tick, and function um, in organisms. Okay, and I kind of already said that before. Up here, when I said the fundamental unit of life is the cell, that's basically the same thing, meaning that your cells is what makes you you. It is why you look the way you look, and it is why you do the things you do. Okay. And the last point is that cells can only come from pre-existing cells. So they just can't come out of nowhere. They can only come from a pre-existing cells. Cells can only come from pre-existing cells. So what that means is that a living organism cannot be just made out of thin air. They don't come out of nowhere. It also means that one cell can only make a new cell of the same type. So for example, humans can only have like a human baby, right? Or horses can only have a horse baby. A bacteria can only make another bacteria baby, okay? And so, and bacteria can't just come out of nowhere. It has to come from another bacteria, like a parent bacteria. And this theory was developed from, there was this famous experiment by Louis Pasteur where he noticed that maggots would become like would just show up on meat, which is just so disgusting. But for many, many years, they thought the maggots just came out of nowhere, out of the air. And so there was an experiment done where there was the meat in a jar and they covered the jar, and there was a meat in a jar and they left it uncovered. And so what would happen is the jar that was covered up did not create maggots, but the jar that was uncovered did create maggots. And that's because flies would go in there and lay their eggs on the meat, and then the maggots were actually baby flies. But nobody saw that. Nobody saw that the flies were landing on the meat. They just saw the maggots all of a sudden. So my point is that from there, that was an experiment that proved that life cannot just be created out of nothing. Okay. And so now it is just like accepted theory that cells can only come from pre-existing cells. Okay, so that's it for this video. I hope you're doing well, and I will see you next time. Bye-bye.